today. So let's get started. It's my pleasure today to introduce our speaker, Sarah Brown. So Sarah Brown is a plant ecologist originally, well, loosely from Texas, we'll say. She did her undergraduate work at Tarleton State University. After graduation, she came to work with us here at Britt for a while, and then went on and worked as a restoration ecology apprentice at The Wilds, which is, if I understand it correctly, is a 10,000 acre conservation park in Ohio that's affiliated with the Columbus Zoo and Aquarium. And I believe somewhere in there during her undergraduate work, Sarah also did a semester in DC and worked as a congressional intern. So she's got a variety of interests and experience. So Sarah is currently a biological field technician with USGS, so the U.S. Geological Survey, and is based out of West Virginia. But this past spring, she just finished up her master's work at James Madison University, and that master's work is what she's going to discuss today with us. So, Sarah, I will let you go ahead. Oh, should have gone to this slide, but I will let you go ahead and share your screen.
Can everyone see it? Yeah. Okay, awesome. Well, cool. Then I'll get started speaking on the project. First off, I want to start off saying thank you to Brooke and to everyone at Britt and for everyone attending this presentation. I am really excited to talk to everyone about the research I completed out of James Madison University. So while there, I explored the plant-pollinator network dynamics of the Little Fork Shale Barren. Before I really get into network dynamics or plant-pollinator networks, I want to first introduce and maybe refamiliarize everyone with ecological networks. So these are the systems of interactions between organisms in their ecosystem. And an ecological network that people are probably pretty familiar with and one that's frequently taught are food webs. These are kind of the systems of the feeders and their food, but they're a really great example of one of the two broad categories that ecological networks fall into. They're an example of an antagonistic network, one in which the two interacting pairs don't really benefit equally. If anything, one partner benefits at the expense of the other. The other category that ecological networks fall into are mutualistic networks. And that's where our plant pollinator networks come into play. These are a system of mutualistic interactions between plant and pollinator communities within a given space. So think of your garden, a forest, those spaces relative as long as the plants and the pollinators are co-occurring and can interact. And we have a method of analyzing and studying these interactions and the relationships using a method of analysis known as network analysis. This is just the application of math and statistics to depict relationships and build interaction networks that look a lot like this. You have your plant species on one side, pollinator species on the other, and lines representing the relationships. So let's get into kind of the nuts and bolts of these interaction networks to better understand kind of the results I'm about to talk about. Plant-pollinator networks are typically shown as a bipartite network, meaning that they're two-parted. You have your plant community on one side and your pollinator community on the other. And what this means is that interactions, any relationships formed between species are only going to be occurring between the two parts of your network. You won't see a line connecting to plant species, for example. So kind of like the interaction network I have here, it's a bipartite network, two-parted. These interaction networks are composed of nodes. And these nodes are the species participating in your network. The relationships between nodes are depicted as links. So these are the lines connecting your species in the network here. At the most basic level, our nodes are described by their degree. So degree is the number of links that a node supports. For example, I've highlighted two plant species in my interaction network. They both have two links connecting them to the pollinator community, meaning that these two plant species have degrees of 2. So interactions in my any plant pollinator network can be displayed one of two ways. So the first is through a binary method. The interaction network I have displayed on the slide here is an example of a binary interaction network where there's just a presence and absence of an interaction. If an interaction is present, you're going to have a link. If it's not present, no link. The second method of depicting interaction in an interaction network is when we consider the frequency of an interaction occurring, essentially giving some weight to the link. The way we depict that weight is through the width of the link, the width of the line, something like this. So this is an example of a weighted interaction network. The thickness of a line corresponds to the frequency of an interaction occurring. Essentially, the thicker the line, the higher the frequency. And it's through network analysis and through these depictions and these interaction networks that we can learn of certain characteristics of our networks. First, we can learn of a network's complexity, how many species are participating in the network and how many interacting pairs are there. We can learn of a network's cohesion, how well connected are our species in our network. We can learn of a network's symmetry. So is there an equal dependence between our communities or even between pairs? And lastly, learn of a network's uniformity. What's the distribution of links within our networks? So thanks to mathematical models that can be applied to real ecological systems, we do have assumptions for mutualistic networks. The first is that mutualistic networks are asymmetrical, meaning that there exists an unequal dependence among partners and between partners. This unequal dependence is best depicted by the figure I have on the slide here. So you have the orange node here having an unequal dependence among its interacting partners. It seems to be more dependent on its lower partner than the upper partner, but there also exists an unequal dependence between our orange node and its partners, showing that it is more dependent on its interacting partners than its partners are on it. This dependence is really depicted by the thickness of these lines. The second assumption is that our mutualistic networks are scale-free, meaning that the probability of a species having a high number of interactions decreases following a truncated power law distribution. Essentially meaning that our mutualistic networks are heterogeneous structures. Most of the species present in the network are not going to be having the same number of interactions. There exists instead, many of the species in a network have very few links, while only a few species have very many links. And lastly, mutualistic networks are nested, meaning that there exists a core of interacting species and peripheral species connecting into that core. On the slide here, I have a figure showing a plant-animal interaction matrix, and it's supposed to show a perfectly nested network. That nestedness denoted by, or that interacting core denoted by the orange square here, with the peripheral species, the squares outside of that orange box connecting into that core. And these assumptions for mutualistic networks provide some support or hint at there's a universal structure present in ecological networks. And these universal structures can hint at a network's robustness to disturbance. So what do I mean by robustness? This is the fraction of species that must be removed to initiate network fragmentation. So how many species can a network lose before you have complete network collapse? Current literature and up until this point, projects have kind of concluded that networks that are fairly complex, heterogeneous, and well-nested have the highest robustness, have a high threshold of number of species they can lose before you have network collapse. But this is if species that are removed at random. And this is because the impact of species loss depends on a species' role in the network. And what do I mean by species role? I'm talking about whether the species removed is a specialist or a generalist. So specialist species are those that have a very narrow range of ecological partners, meaning that they have a very low connection into the network. While generalist species are those that have a very broad range of ecological partners, making them fairly highly connected. And it's that difference in connection that can translate into differences in the impact of their loss. So I'm going to show you what I mean by that on this slide here. I'm showing you an interaction network where I've removed one of the more specialist species from the network. If the network is fairly complex, heterogeneous, and well-nested, you would ideally see the network absorb the loss of the specialist species and still retain its overall network function. Story's a bit different with the loss of a generalist species, so that same network, but this time I've removed my most connected species. And you can probably already tell that there's a larger impact with the loss of such a well-connected species. Two of the pollinators in the pollinator community have had their foraging resource cut in half, and another pollinator has had their foraging resource removed entirely. So this is a really great example of how primary extinctions of a general species can kind of have a cascading effect, leading to secondary extinctions and even eventual network collapse. And it's through network analysis that we can learn and become aware of these roles and kind of the hierarchy in our ecosystem. And when we're aware of these roles and hierarchy, we can better target some of our conservation efforts. But all this isn't to say that networks don't change. Species, they're not fixed entities. Up until this point, I've kind of been talking about networks as if they are fixed entities, when they are really actually very dynamic. So to kind of prove my point or to talk about it a little bit more, let's say you go out to your garden, you complete a survey of your flowers to see what insects are visiting. You construct an interaction matrix or interaction network at time X. You come back a day later, a week later, a month later, conduct the same sort of experiment, make the same observations, and build another interaction matrix. You will find that when you compare your networks, you're going to see that they're not going to be equal to each other. And this is because the interactions formed between plants and pollinators are very dynamic, known to change across time and even space. So projects that have now been exploring plant-pollinator network questions are now considering this inherent dynamic nature on the formation of these networks, on the formation of these interactions. And that dissimilarity between networks maybe at the start of a season and the end of a season is described as interaction turnover, which is described by the formula provided below on the slide. So interaction turnover is the sum of two turnover components. It's interaction rewiring and species turnover. So interaction rewiring is a situation where you have your one network, you have your species composed, interacting in one way. When you compare to the next network, maybe in your season, you'll find that those same species are present, but interacting in different ways. Species turnover is a little bit more self-explanatory. You have your composition of species in one network, you compare to the next, and you'll find your composition has changed. There's different species present and therefore different interactions occurring. Interaction rewiring and species turnover are happening simultaneously in your ecosystem. It is not one or the other, it's not a sequential situation, but there are discrepancies of which one is kind of the driving force of the overall change and turnover in your ecosystem. And when we're aware of the driving force for overall change, as well as the rate of the change, we become more aware of the flexibility of interactions, how the networks and interactions form and dissolve, and be more aware of that dynamic nature of our ecosystem. So I've already kind of been touching on it this whole time, but I do feel that network analysis and the construction of plant-pollinator networks can serve a real tool in conservation, namely that it can provide a lot of context. We become informed of the patterns of interactions between species, between pairs, and even across the communities. And when we're informed of these patterns, we can better predict the impacts of ecological change on this functional aspect of our ecosystem. What happens when a species is really removed from the ecosystem? What happens when a species is added? What if the populations change in their distribution? When we have this information to make these predictions, we are better able to target some conservation efforts and be prepared for some of these changes. And an ecosystem that could really use a lot of context are shale barrens, so the other part of my title. These are pocket ecosystems found among the temperate forests of central Appalachia. They are typically found on mountainsides at a minimum grade of 20 degrees. So not a lot of chance for water retention in these spaces, making them very dry. Along with that steep grade, they're normally found at, on mountains with aspects of south by southeast, meaning they receive a lot of direct sunlight, only adding to this hot, dry ecosystem. Due to the slope, they also have very little organic matter accumulating on the space. So a lot of times any trees that can survive, any shrubs, and the herbaceous species are growing out of essentially shale rock fragments. An overall not hospitable sounding environment. But these shale barrens support a very unique flora. Most of the species found within shale barrens are fairly characteristic for the spaces, and they grow best in these environments. And shale barrens are known to have 18 species endemic to this ecosystem. For reference of, since they don't really cover a lot of space, this is actually a fairly high number. For reference, the state of Virginia is known to have 5 endemic species. Shale barrens also have a relatively unexplored insect fauna. So I could probably count on my hands the number of papers that discuss the insect community of shale barrens. So a goal of my project was to kind of add to the pool of information for at least the pollinators of the shale barren, let alone the insect community in general. And lastly, shale barrens are globally uncommon. So on the slide here, I have the global distribution of shale barrens. They are unique to Central Appalachia. So there's just this kind of gem in the area, an ecosystem that can support such unique species and organisms that we just don't know enough about. Currently, conservation plans for shale barrens are very hands-off, which kind of paints us into a corner when we do need to apply any conservation efforts to a shale barren. We just don't know what to do. And that pulls me to the star of the show, the Little Fork Shale Barren, where I did all the research for my master's thesis. This shale barren is one of 7 in the southeastern portion of Pendleton County, West Virginia. This was about an hour due west of James Madison University, so I typically just drove over to conduct my field work. This shale barren is unique to the other 6 in the area in that it is located on private property. It's actually on the property of the Sugar Grove Naval Base and Research Station, meaning that there's kind of infrastructure around it. There's accessibility, there are roads, and I have access to it through permission. And it's also had a history of monitoring of its vegetation. So in 1992, a fixed monitoring grid was established by the West Virginia Department of Natural Resources in the hopes of long-term monitoring for the space. That fixed monitoring grid was used by a master's student out of Marshall University, where he conducted a descriptive study of the site initially in 1994. And he found that there existed 6 populations of endemic plant species within this space, only adding to the importance of this shale barren. My thesis advisor, Dr. Conley K. McMullen, also has conducted some of his own studies there exploring pollination ecology and has conducted studies as recently as 2014. So there's still a really immediate partnership with him and security at the base. So when I was entering my master's thesis, I kind of faced the question of why not conduct a project here when there was so much history and so much infrastructure to study such a unique space. And this gets me into my study objectives for my thesis. My first objective was to assess the vegetation community and compare current community, that being 2020, to the descriptive vegetation study that happened in 1994. The next was outline the structures of plant pollinator networks within the space. I tried to quantify the full growing season, what were the relationships formed within that space. And lastly, I wanted to examine the scale of within season interaction variation for the site. This was within season interaction turnover. I'm going to start off talking about how I assess the vegetation community. So since I was trying to make comparisons between my 2020 results and the 1994 study, I did have to follow methods provided by the student out of Marshall University. I conducted bi-monthly surveys starting August 26th through October 10th of 2020. And these surveys were conducted within a 200 meter by 50 meter section of the fixed monitoring grid. This is everyone's first view of the full grid. So for reference, the ridge line runs parallel to the top of this grid. Within the 200 meter by 50 meter section, I picked, following the footsteps of the student prior, I selected specific cells for my vegetation surveys denoted by the green boxes here. These green boxes are actually 10 meter by 10 meter cells. So there's a full grid and then there are each individual cells, and it's within these cells that I randomly kind of divided them up and randomly selected a place where I put down my 1 meter by 1 meter plot. And it's within this 1 meter by 1 meter plot that I identified all vascular plants within that space and estimated their cover. But I didn't stop there. Actually, I also included how much of the plot was actually bare ground showing rock fragments and how much of the space was bare ground but dense leaf litter. You might be wondering why I included those categories. So I'm going to show you the range of what I saw within these plots. Sometimes plots were bare. Sometimes it was just bare rock fragments, something that actually is somewhat expected for a shale barren. Maybe just like the rocks fragments, it's a typical substrate. The other end of the spectrum was a well-vegetated space, a lot of characterized and grasses, but there were areas void of plant matter, but there was no direct access to the substrate. It was just dense leaf litter instead. So essentially the goal was just to make sure that I quantified the full space of my plots and didn't leave anything out when I was collecting my data. With this data, I went about trying to assess how much of the shale barren was vegetated versus non-vegetated. Shale barrens are described to be sparsely vegetated. Is that true for the Little Fork Shale Barren? Next, I identified the number of species found only in 1994, found only in 2020, or the species found in both surveys. And then I identified my 2020 most prevalent species. To do this, I considered the mean estimate cover and their importance values. That importance value was the average of their mean relative frequency and mean relative cover. This formula provided to me by the Marshall University student. And then lastly, make my comparisons with the 1994 study. So the metrics I was comparing was species richness per cell, species diversity per cell, and then equitability per cell, which is essentially a measure of evenness, calculated as the species diversity divided by the max possible species diversity. Now to get into the result of this part of the project. I found that vegetation covered about 45% of the shale barren, so that still meets a sparsely vegetated status. The area hasn't turned into a lawn or something. I found 69 vascular plant species through my surveys. 51 of those species were found only in 2020. 13 of the species found in both surveys, 1994 and 2020, and that left 5 species found only in 1994. My title for most prevalent plant species went to Carex pennsylvanica, that's the Pennsylvania sedge. Not a totally surprising result, actually. This is a very characteristic shale barren species known to dominate the kind of the open spaces of shale barrens. And this plant was the most dominant species in 1994. Though its level of dominance has definitely decreased, likely just because there seems to be an increase in the number of plant species found through my surveys, just found in the site. The second most prevalent species I found for the site, though, was a little bit more surprising. This went to the plantain leaf pussy toes. This is Antenaria plantagifolia, not a shale barren characteristic species. If anything, it's a general woodland species. It is also one of the 51 species that I found only in 2020. So this hints at maybe some changes in the site that should be explored further to understand a little bit more of how general woodland species could be more competitive than species better adapted for shale barren spaces. Now to get into the comparisons. The data for my results, as well as the 1994 study, were not normally distributed, so comparisons were made using a Wilcox ranked sign test, and the results of those studies, or the results of those tests, are shown at the top of these box plots, with stars representing a significant difference and NS representing no significant difference. You'll find each of the metrics shown kind of running across with species richness per cell on the left, diversity in the middle, and equitability on the right. And what you might be able to note here is that we have seen, or I found that there has been a significant increase in the species richness per cell and species diversity per cell for the site, while there seemed to have been no significant difference in equitability for the space. Maybe they're just like a contraction in the spread of the values. So what's my assessment? What is the assessment for the vegetation community of the shale barren? Well, it's still a sparsely vegetated site. It hasn't turned into a lawn. It hasn't been overtaken by trees, by the surrounding temperate forests, but change has occurred. The site now supports a richer and more diverse plant community, something worth noting. It's a dynamic space. Change is occurring. Now on to the second objective of my thesis, outlining the structures of plant pollinator networks. To do this, first, I wanted to identify phenologically accurate networks. Next, calculate network metrics. And then identify my highly connected plant species and key pollinator groups. What are my most connected, most relevant species? To do this, I utilized the full grid, and I wanted to cover as much of the growing season of the shale barren as possible. So I conducted 2 types of surveys starting April 21st all the way to September 22nd. This was of 2020 and about 6 months worth of sampling. The first survey I conducted, or one of the 2 types of surveys I conducted, were flowering inventories. This consisted of just presence and absence of flowers, marking to see where they were and what species were in flower. My second type of surveys were pollinator surveys. So this was identifying flower visitors and then getting account of the visitation frequency of those visitors. So I do want to point out that up until this point in the PowerPoint, I have been saying pollinator, and I do plan to continue saying pollinator through the presentation, but I do want to point out that I really was surveying for flower visitors and that these flower visitors have different capabilities as pollinators. But for ease of speech and just clarity, I'm going to continue saying pollinators. So my surveys were conducted within the full space of the grid. This is 16,000 meters squared. This grid consists of 160 10 meter by 10 meter cells. And since I was covering also the full, as much of the growing season of the shale barren as possible, I divided that time frame into 10-day monitoring periods. You can kind of think of it as cycles of sampling, with day one of a cycle being marked by my flowering inventories. Flowering inventories just consisted of me visiting every single cell of the grid, marking if flowers were present, and then identifying those flowers, coming to a result after day one that looks like this. Green represents that flowers are present. The rest of the monitoring period was then dedicated to pollinator surveys. Though those pollinator surveys were typically started and completed by day 2 or 3. I didn't want to be too far removed from my flowering inventories. I did not, however, conduct pollinator surveys in every single cell that had flowers in it. Due to COVID, I was working alone, and I just wanted to make sure I focused my effort where I was going to be getting results. So I added an extra criteria when it came to my surveying, that when I saw flowers were present in a cell, I then kind of re-examined the space to see if the cover of those flowers was at least 5% of the space. If the cell met that criteria, I could then say pollinator survey should occur within the cell. To distinguish or at least designate the specific location of pollinator surveys, I identified flowering hotspots within my cells. So these were areas where there was just a relatively high density of flowers from either a single species or 2 or more species in flower within the same kind of space. It was once I identified those flowering hotspots, I could lay down a 1 meter by 1 meter grid, observe that a plot, 1 meter by 1 meter plot, observe the plot for 10 minutes for something that looks like this. This was a flower visitor, a pollinator, visiting the reproductive parts of a flower. When I saw something like this occur, I identified the pollinator, identified the plant being visited, and the number of visitations completed by the pollinator. It was with this data that I can move on to identify my phenologically accurate networks. And you might be wondering why I might want to do that. Can't I just pool all the data together? Well, I would really rather not pull all the data together, actually, because when you arbitrarily pull data, you're going to be getting forbidden links in your network. So forbidden links occur when you pool data, observation data to form a network, and you include species in your network that naturally would never interact. Why 2 species might not interact could be that they aren't active at the same time. Spring species aren't interacting with summer species. They don't occur within the same space, and their abundances don't promote their interaction. A bee finding the flower of a single plant within its full foraging range is just so unlikely. A lot of projects that are looking to now explore questions in plant pollinator networks that they try to achieve these phenologically accurate networks, they focus on plant phenology for the construction of their networks. This plant phenology being that the time frame when a plant is producing its flowers. And why they would want to do that is because plant phenology is typically a constraining force in the formation of networks. Plants may produce flowers for only a few weeks, while pollinators could be out for several months. So this is something I want to do for my own project. I also focus on plant phenology for the construction of my phenologically accurate networks. And so what I did was I used a Spearman correlation to see if my monitoring periods could be correlated based off the species in flower during that time. If monitoring periods were significantly correlated based off of their species in flower, then they must form a unit, and then I could pool that information together to form my network. Once I identified my phenologically accurate networks, I can move on with calculating my network metrics. Though I say calculate, I did not pull out a calculator and type in some numbers. I instead used the bipartite package in R to find my values. The first metric I wanted to identify for each of my networks was to figure out the degree distribution of each network, ideally looking to see if my networks were scale-free, where most of the species had very few interactions and a few species had very many interactions. The next was identify the interaction strength and symmetry. Again, the assumption that mutualistic networks should be asymmetrical. I want to know kind of the direction of that asymmetry. Were plants more dependent on pollinators or vice versa? Next was to calculate the connectance of each of the networks. So this is a measure of the complexity We want to understand complexity for maybe getting assessment to predict the robustness of each of our networks. And connectance was calculated as the total number of observed pairwise interactions divided by the possible number of pairwise interactions. And lastly, determine the nestedness, the level of nestedness for each of my networks. How well cohesive were these networks present in the shale barren? Again, trying to see if that expectation, the nestedness of mutualistic networks, was present in this space. For identifying my most relevant plant and pollinator, I went about using species strength to identify the most relevant species. Species strength was the sum of dependencies for each node. The idea being that a node and a network with a higher number of other species dependent on it must make it more relevant. So let's get into some of the results of this part of the project. These are just a few of the thousands of pictures I took out in the field, some of my favorites, but I did get into kind of wildlife photography through this work. I completed 16 monitoring periods through my season that consisted of 530 pollinator surveys, about 88, 89 hours looking at plants for pollinators to visit. I found that 42 plants and 85 pollinators comprised my full shale barren system. So this is spring to the end of, to the summer. That's 325 unique pairwise interactions. And I identified 3 phenologically accurate networks. Though this part was a little less cut and dry than I was expecting, there just exists a really strong overlap in plant phenology for the shale barren, which is objectively great. That means there's no periods of lack of foraging resources for pollinators, but just didn't help me when it came to building networks. I just still couldn't confidently pool all my data together since spring species are not going to be interacting with summer species. So I still identified a spring network and a summer network. The spring network marks the time frame of the start of the sampling season, so the end of April to the start of June. And the summer covers the time frame of the start of July through the end of the sampling season. That's the end of September. But it was the middle part, it was the time frame in between those that kind of got a little tricky. So what I did instead was establish more of a transitional network. This is a shorter time frame, only covers the start of June to the start of July, and it overlaps with my spring and summer networks. The species included in this transitional network are those that really had the long-term phenologies. These species were ones that kind of flowered in the summer and even continued well into the summer. So it's with these phenological networks, I could then calculate my network metrics. So let's look at those visuals I was able to make. I will say that this slide gets a little overwhelming, but I'll do my best to explain everything and make it as clear as possible. So I'll start off showing my spring network. Along the bottom, you'll find the plant species in black squares. The pollinator species are running along the top, and they are color-coded based off their pollinator group. So to explain which group is which, gray represents bees, blue represents butterflies, orange represents flies. There's a little bit of red over here in the corner that represents ants. You see kind of some sections of green, those are wasps. And then lastly, there's a little sliver of purple that are beetles. This is a network that you are probably noticing that some of the boxes, some of the nodes are varying in size, and that's because the size of a box corresponds to a species degree. So species that support a higher number of links, they have a higher degree, are going to have a larger box. An easy way to visualize which ones are really well connected in the network. This is also a weighted network, so I considered the frequency of interactions, hence why you see some differences in the width of links in the network. Again, saying this is just to tell you that some interactions between two species are much stronger than others. I also color-coded the interactions based off of the pollinator group that is connecting to that link. So you're probably able to really see which group of pollinators is really dictating the linkage of the network. And you're probably seeing a lot of gray right now. With all that explained, I'm gonna put onto the slide my transitional period and my summer network as well. Again, you're probably seeing a lot of gray, and you can still be able to take note of which species are really well connected in this space. For all three networks, I found that they followed that truncated power law distribution. They're all scale-free, making them following and meeting that expectation for mutualistic networks. All three also had a fairly had connectance levels that ranged from like 0.1 to 0.15, which when you know that connectance ranges from 0 to 1, you may think is objectively low. But when I did some digging in the literature, I found that these levels of connectance, this level of complexity, is actually very comparable to other ecosystems. And for all three networks, I found that they are fairly nested. They have nestedness can range from, again, 0 to 1, and most were hovering around 0.85 to 0.9. So there exists that core of interacting species with peripheral species connecting into the core. And lastly, they are all asymmetrical, and for all three networks, I found that the pollinator community is more dependent on the plant community, likely just because there are just so much fewer plant species to choose from for the pollinators. Now on to my most important, most well-connected plants and pollinators. For the plant species, I found that in the spring, the early low-bush blueberry, Vaccinium pallidum, was the most well-connected. The narrow-leaf bluet, that's Houstonia tenuifolia, was the most relevant species in the transition period. But again, to reemphasize that these species were ones that had pretty long phenologies, and this species was no exception. Narrow-leaf bluet was present for 15 of my 16 monitoring periods. Its relevance was just highest in this transition period. And then lastly, the elm-leaf goldenrod was my most relevant species for the summer. Something to definitely point out, since the summer network really covers a long time frame for the growing season of the Shale Barren, maybe a point of interest if conservation efforts were to begin at the Shale Barren. Moving on to my pollinator morphospecies, this is less of this morphospecies is important at this time frame and more of a ranking system, since these pollinators were all present for each of my networks. My most relevant pollinator morphospecies was the small carpenter bee. These are bees within the genus Ceratina. Next, most relevant were the metallic sweat bees. These are bees within the genus Lasioglossum, subgenus Dilectus. And my third most relevant species was one I could identify as species. This is the golden sweat bee, Agochlorella aureta. So it's really great that I was able to identify these important and highly connected species and morphospecies, because this can, if conservation efforts were going to begin or occur in the future, this is maybe a jumping off point that efforts could focus in their efforts. What species are going to be really pivotal in protecting the space? So what are some of the conclusions that I can make with the plant pollinator networks of the Little Fork Shale Barren? First, the networks are cohesive and heterogeneous structures that have complexities comparable to networks found in other ecosystems. So providing more support that there exists these universal network structures across ecosystems. And that the pollinator community is more dependent on the plant community. Something I really want to emphasize, since we just found evidence that the plant community has changed and will likely continue to change. What does that change mean for the pollinator community? So to continue with the spirit of change, I'm going to move on to talking about how I examine within season interaction turnover. So the first sub goal of this objective was to just identify within season interaction turnover. Is it high or is it low? Interaction turnover can range from 0 to 1. Where does the Shale Barren fall within those numbers? And then I wanted to find out which component really dictates interaction turnover. Is it species turnover or interaction rewiring? The second sub-objective was to identify any relationship between pollinator abundance and interaction turnover. So this question came about from a 2017 study conducted by Caradonna et al. That found that species abundance closely predicts interaction turnover. They're closely tied. But having conducted pollinator fieldwork before and seen how it can fluctuate day to day, I wanted to see if those fluctuations in pollinator abundance can be reflected in interaction turnover values. Now to go on, like how did I answer any of these questions? Using the data I collected through my field season, so following the same methods that I described earlier for constructing my networks, I then used that data to identify and calculate interaction turnover values. I calculated these values using the beta link package in R. So I didn't pull out a calculator again and just type in numbers. Once I calculated these values, I then compared interaction rewiring and species turnover using a two-tailed t-test. To identify the relationship between pollinator abundance and interaction turnover, this required me to create two types of probability interaction models. The first model considered only species phenology. This was a null model. Two species have to be active at the same time to interact as a baseline requirement for interactions to form. The other model considered species abundance and species phenology. And this was the model that could help me confirm whether species abundance also predicts interaction turnover values in the shale barren ecosystem. From these models, I completed 1,000 iterations, and from those iterations of essentially 1,000 iterations of different networks, I calculated their turnover values, essentially producing some simulation turnover values. With those simulated values, I then compared them to my observed values, calculating a standard effect size, the idea being that any standard effect size value falling within a 95% confidence interval means that the model predicted my turnover values. And if the model predicted the turnover values, those ecological constraints included in the model therefore predict turnover values. This was a really important part of this objective for this part of my project. And once I was able to complete that and see if species turnover was closely tied, or species abundance was closely tied to turnover values, I could then plot turnover values and then pollinator abundance through my season. So with all that out of the way, we can get into some of my results. Again, I completed 16 monitoring periods. So that meant I had 16 fine scale networks through my season. And these fine scale networks really ranged in size. So as some were as small as comprising only 11 pairwise interactions, and some ranged to be as large as having 55 pairwise interactions. But the data was fairly normally distributed, and all values I found on average hovered around a 0.75 interaction turnover value. So on the slide, you're seeing box plots representing interaction turnover, interaction rewiring, and species turnover. Through my two-tailed t-test, I found that interaction rewiring significantly contributed more to the overall change in my system, to the overall turnover value, than species turnover, meaning that the same species were present in my networks, but they're interacting in different ways. Moving on to my models. So on the slide, I'm showing you the results of my null model. I have plotted out my standard effect size values for interaction turnover, interaction rewiring, and species turnover, with values that were not predicted by the model in gray and values that were predicted by the model in black. And you're noticing probably a lot of gray dots in this model. I found that species phenology alone does not predict interaction turnover values, a result very similar to the 2017 study I'm kind of following. Moving on to the model that considers species abundance and phenology, I found that species interaction turnover and interaction rewiring were well predicted by the model, hinting that species abundance is closely tied to predicting interaction turnover. Situation was a little bit different, a little bit more on the fence with species turnover for this model, but this really just implies that there's an ecological constraint that I haven't considered and isn't included in the model that better predicts this value. But I got the green light to proceed with plotting out my interaction turnover with my pollinator abundance through the season. So let's see how that plot looked. Along the bottom, you'll find monitoring periods, the start of the season, the first monitoring period in the left corner, the last monitoring period in the bottom right, interaction turnover values denoted on the right, pollinator abundance on the left. The black line is the plotting of my pollinator abundance, and the red line is the plotting of my interaction turnover values. And in the amount of me talking, you probably noticed that there's sort of a reflective pattern at the later half of my monitoring periods, with steep declines in pollinator abundance being matched by steep spikes, or essentially spikes in interaction turnover. Not an entirely conclusive result, but at least can point to the fact that there does exist some kind of relationship between these values. So some of my conclusions based off of this section of my thesis. I found that within the shale barren, there exists a high level of change within the season, that interactions are forming and dissolving fairly rapidly, and that this form and disassembly of networks is really dictated by the same species interacting in different ways, essential flexibility in their interacting capabilities. And I found evidence that interaction turnover does seem to be sensitive to the abundance of the pollinator community, which is really more of a sampling concern. Any future projects that may look into interaction turnover for a space should really take into consideration how many pollinators they may have seen on the day of sampling. To say that a space is experiencing a complete turnover when you saw just a lot fewer pollinators may be a little misleading. But this doesn't ignore the fact that there's a lingering question of like what are the factors that may cause pollinator abundance to fluctuate, since those factors do seem to have a role in the formation and disassembly of networks within a space. So what is the overall assessment of the Little Fork Shale Barren? I've covered a lot of sections of the project. What was I able to overall conclude for the space? Well, it's still what you may expect a shale barren to look like. It's sparsely vegetated, but that sparse vegetation, that barren aspect does not equal lack of activity and diversity. It is a vibrant space, one that we weren't entirely expecting, especially based off of how literature tends to describe them. Networks present within the shale barrens follow typical network structures and expectations that are found in other ecosystems, providing a little bit more credence that there exist these universal network structures that we can apply to other ecosystems. There is a hint of interaction flexibility among species for the shale barrens, since I found that interaction rewiring is the driving force for overall turnover. Something great to note that it's just a matter of the same species interacting in different ways. And lastly, it's just overall dynamic space. It has changed in the past decades. It changes throughout a season. And honestly, it's going to be likely changing into the future. Which gets me to the last slide of my presentation. What is the future of the Little Fork Shale Barren? Which I really should say, what are the lingering questions for the Little Fork Shale Barren? First, I was left wondering, what are the forces maintaining the current network? So you might be wondering what forces I'm talking about, but are the current networks something that we are, that's meeting expectations? Are they being maintained by the composition of the communities, plant or pollinator? The abundances of species within the communities, their distribution, as well as just like environmental factors such as weather or water within the space. What are maintaining the current state of the networks? Because when we understand what maintains the current networks, we can understand how change might affect the structures of the networks. I can't make too many conclusive decisions about how these networks may change when things are changed around them. That requires other data manipulation. So I hope maybe in the future, I can take some of the data that I collected from the season and do additional data manipulation to better understand how change will affect these networks. So with that all said, I just want to acknowledge I had help with my committee, but also point out that my advisor was Dr. Conley K. McMullen, who was really the greatest support for this project. He really helped me push my limits. I want to send a thank you out to Anna Harnick. She was some of my field help for the end of the season, despite COVID happening. We did get some permission. Thank you to the security base, the Sugar Grove Naval Base, their whole security force. They saw me for 6 months, and I think by the end, they were just wondering why I don't live there. And then lastly, thank you to PJ Harmon. He is the botanist out of West Virginia DNR and the person who established the fixed monitoring plot. Without him, I wouldn't have done this project. Oh, and then lastly, thank you to all my funding sources for the project. Texas Hill Country Branch for the American Association of University Women, Garden Club of America, and JMU Department of Biology.